Hello, welcome to part 2, immunity. So last lecture was all about innate immunity, which is non-specific, meaning it doesn't matter what the pathogen is, your body will automatically respond to an invader, and you have these built-in mechanisms of protection. Today is all about adaptive immunity. And so this is how we specifically target specific pathogens. Mainly, I guess one of our main focus is formation of antibodies and how antibodies are able to target very specifically antigen of pathogens. And we will also look at cell mediated, which is T cells that will also directly target a pathogen. Okay, so let's get started. So, this type of immunity, adaptive or specific, is built over time, right? It takes an exposure to the pathogen's antigens in order to make antibodies. So, naturally, as you're exposed to pathogens over the course of your childhood and young adulthood, and even into adulthood, you will make antibodies. But if you have a vaccine, you don't have to be exposed to the actual pathogen, meaning you don't have to have the risk of getting sick. You're able to make antibodies from having just the antigen, not the entire pathogen. It does require you to have lymphocytes, T and B cells, okay? And when you hear T and B cells or lymphocytes, you should be thinking lymph nodes. The T and the B cells are most concentrated in your lymph nodes. And so we kind of think of that as being the site of your adaptive immunity, at least humoral. You have two branches of adaptive immunity, humoral and which is for antibodies, and then cell mediated. Okay. So often adaptive and innate immunities overlap, right? They work together. So inflammation and the complement system are used in both types of immunity, right? As the cells of the adaptive immune system are working, the innate responses are also working to control and stop the infection. So the life of a B cell, remember that B cells start out in the bone marrow and they will stay in the bone marrow and mature. And then your mature B cell, what's happening in between being immature and mature is it's gaining its specific receptor, its unique receptor, and then it's going to travel to the lymphatic system, primarily your lymph nodes. It does that through the blood. Some will always circulate in your blood, but the vast majority are going to hang out in your lymph nodes. Okay, and then if you have an antigen, a foreign particle, typically a pathogen, then that, if it finds the matching B cell, because you have hundreds of thousands, if not millions, of B cells, each with a unique receptor, waiting for a situation where there's something that matches to it. So essentially, your body has already prepared for any and all pathogens. Every single combination of an antigen, those are your B cells. Okay, so each B cell has its own unique receptor. waiting in case you ever are exposed to the corresponding antigen. So in this flowchart, we're saying that, let's say you have a B cell and it does come in contact with the matching corresponding antigen, then that's going to activate your B cell. And as long as you have a helper T cell to come along and tell the activated B cell, yes, go ahead, right? So let's say you have the helper T cell. It tells your B cell, yep, go ahead. Then you're going to make a whole bunch of identical copies, which we call clones. So this is called clonal expansion, and that's going to lead to your plasma cells and your memory cells. So what happens is you make this huge army, right? These clone army, like Star Wars, right? But not evil. And so you have this army of clones of B cells that have the receptors for that specific antigen, right? And then, I don't know if it's half, but a portion will become plasma cells, which just secrete tons and tons of antibodies, which will be specific to bind to that antigen that activated it. Another portion of that army will become memory cells. They sit around essentially for the rest of your life in case you ever have an exposure to that same antigen again in the future. And this is the reason we have vaccines. Vaccines are to give you only the antigen so that you activate a B cell and you go through this process of making antibodies. But the goal of the vaccine is to make these memory cells so that if you ever actually get contact with the actual pathogen, you will immediately secrete tons of antibodies and never even have to get sick. So that's the idea and how vaccines actually work. Okay, so one more time. B cells, they begin in the stem cell, as a stem cell in the bone marrow. They stay in the bone marrow, and you're going to have, let's say, millions of unique B cells. They're unique because they each have their own unique receptor, and the idea there is in case you come in contact with an antigen that matches it. So your body wants to go through and make every possible combination, and then we have to make sure that it doesn't match any of your own receptors. And so that's also part of this maturation. So during this step, this is the step that we call clonal selection. We're going to select for B cells that are unique, meaning that they are not going to react with us, okay? And then each B cell is unique from one another so that you have every possible combination so you're prepared for any pathogen or any antigen that comes your way. Once that check and balance of making sure it's not going to react with your own cells, once that's completed, it travels to the lymph nodes primarily, and waits. And then one day, let's say one of those B cells is introduced to or comes in contact with its corresponding antigen, that's going to activate it. It's going to sound the alarm, right? Like, oh, I got an intruder. I found one, and then it's going to make that clonal expansion or that army of clones, and then part of those become plasma cells, which secrete tons of antibodies into your blood so that it can spread throughout and get any more of those antigens that just want to bind to it and make it easy for phagocytes to then come pick it up. And also, think about if you had a bacterial cell which is covered in antigen, you cover all of its antigens with these antibodies, and it can't go anywhere, and it can't do anything. And it's just a sitting target now for a phagocyte. Same idea with a virus. If you cover up all the receptors on the virus, it won't be able to get inside of your cells, and it won't be able to infect the cell. Okay. And then part of that clonal expansion army will become memory cells to give you your lifelong immunity. And that's how, or that's the goal, and that is how vaccines work. Okay. So as I said, you have your B cell, and its receptors are antibodies, and the end of the antibody will be unique and specific, and only one type of antigen will interact with it. And one day you come in contact with that antigen, and it binds and it activates this B cell, and then it makes lots and lots and lots and lots of identical copies. And some of those copies become plasma cells, which just secrete antibodies into the body blood, right? So that you can catch all of your invader if there happens to be more, right? And then you have some of those become memory cells to protect you in case that antigen ever appears again. And typically, like if you have an infection, this antigen will be attached to the actual pathogen, right? But we only show the antigen because that's the part, that is the only part that's required for this process, which is why vaccines can work. But again, typically in the body, the antigen would be still attached to the microbe, the pathogen. Okay, so B lymphocytes are B cells, okay? They work by antibody-mediated immunity, or humoral, Humoral, humoral. And at the very end of this lecture, I have a messy handwritten little chart for you to help you. Okay, so B cells have receptors, which I've talked about and showed you on the last slide, and those receptors will bind with their corresponding and specific antigen. So what are antigens? I've been talking about antigens a bunch, and I haven't explained what they are. So they are proteins. Typically, it could be a carbohydrate, a glycoprotein, a glyco, any combination of protein, carbohydrate, and fat, typically just carbohydrate and protein. And these are also on the surfaces, or which we call receptors, and we also call antigen, but they're on the surfaces of your viruses, your bacteria, and then sometimes things that you're allergic to is really your body misidentifying a particle as a pathogen. So we said allergens, but that really shouldn't be the case, right? You shouldn't be having an immune response to dust or pollen or any of these other things that cause allergies. That's a misfunction of the immune system. Okay. So after recognition of the matching antigen, B cells will divide, and that's clonal expansion. And again, this requires a helper T cell. Some of these clones, these exact copies of your B cell, will become plasma cells, and others become memory cells. Okay, so now let's talk about the interaction between your antibody and your antigen, and then also the structure of an antibody. Okay, so as I mentioned, the plasma cells will secrete antibodies. Antibodies have these two main parts to them. We have it color-coded here to show this, what we call the heavy chain, and in green we call the light chain, because here it's only held together right here and here and here. So these can become detached, such as during ultracentrifugation. And so when you centrifuge it down, you'll get two bands or regions, the small ones and the long ones. Okay. So, anyways, back to how this antibody functions. We call the very top portion, which includes the blue and the green, labeled VH and VL. Okay, VH for heavy chain, L for light chain. The V stands for variable. So this is where the interaction between the antibody and the antigen takes place. So notice that the VH and the VL both interact with the antigen, which is this orange rectangle, square, diamond. Okay. And so we call it the variable region because every type of antibody is going to have its own unique binding site to bind with a different antigen. Okay. Then this whole bottom region, all of the ones labeled C stands for constant region. Okay. So all of the rest of this antibody will be the same for every single type of antibody you have. So only the variable region changes from one antibody to the next to bind to a specific antigen. Okay, so each antibody has 4 polypeptide chains, 2 heavy, 1, 2, and 2 light, 1, 2. And the most important part of this of antibody structure is knowing the variable region versus the constant region. So only the variable region is able to bind to antigens. Okay. So you have a huge diversity. There's about, 100 million trillion antibodies in your body. This allows you to attack a few million different types of antigen. Variety of all these types of antibodies are made by combining heavy and light chains. So that variable region, one part of the variable region is heavy and the other part is light, and you will change the amino acid sequence on this to get all sorts of different combinations. Okay, so let me say that again. So remember that the antibody is a protein, immunoglobulin. So it's a globular protein, okay? And you want to remember that it's a protein because it'll help you remember that this region here, the variable region that's binding to antigen, it's actually amino acid sequences. And these amino acid sequences interact with the amino acid sequences on the antigen. Okay, and so that's why by creating all these different combinations of amino acids, you're able to get all these different types of antibodies. Okay, and we know that the functions of antibodies are to stick to the antigen so that phagocytes can find it easier, grab onto it easier, and eat it up and get rid of it. So we call this opsonization, enhancing phagocytosis. Okay, so you need to know that opsonization enhances phagocytosis, but you also need to know how. And I just explained that, right? So the antibodies are going to bind to the antigen, and that's going to make it easier for the phagocyte to find, bind, and eat up whatever it caught, right? So that could have been loose antigen, or it could have been antigen attached to the pathogen. All right, so it also causes clumping. So let's say you do have just antigen, um, it'll, no, let me start over. Clumping. So you have um, a couple of bacteria or a couple of viruses, and the antibodies can help clump them together, which will also end up leading to phagocytosis. Okay, and I think I'll have a picture to show you for clumping. If not, I'll try to post it as an announcement to help make that more clear. Okay, and then the third function is to activate the complement system. So for that, you would need to go back to the last lecture where I explained how the complement system works, and I gave an example of a pathway where antibodies are sticking to a microbe, and then if you have two antibodies right next to each other, then the protein in your blood that's always circulating, the complement protein, will bind to those two adjacent antibodies, and that will cause it to split in half, and you start this cascade called the complement system. Okay, so go back and look at that. Okay, memory cells, I told you, in addition to the plasma cells that secrete antibodies, you also have memory cells. Memory cells sit around, wait in case you ever come in contact with that antigen again. So the first one that led to the formation of memory cells was called the primary response. Any exposure after that, any future exposure that causes your memory cells to become active is called a secondary response. And this is going to be very rapid because you already went through the whole process. Now you're ready to just secrete antibodies. But it also turns out that you'll secrete a much larger amount of antibodies and for a longer amount of time. So your body makes sure that this pathogen is not a problem. And you won't experience any symptoms when this happens. And so this is what it looks like. So your primary response, right, it takes about a week, sometimes a little bit longer, to make antibodies, right? So you get to about week 1 of being sick, okay? At this point, you've experienced symptoms. You feel awful, typically, depending on how severe or mild the illness is. And then you start to secrete antibodies, right? And this would be your plasma cells. Your plasma B cells are secreting antibody. And this is what that all looks like, okay? And after week 4, you slow way down and you produce way fewer antibodies, okay? However, if you ever come in contact with that same antigen again in the future, immediately, right, within a day, your memory B cells will secrete antibody. And look how extremely high the level of antibody secretion is, right? So along this axis would be the amount of antibodies per milliliter of blood, something like that, or serum. And it's so many more than you ever made in the first place, right? The most you made before was here, about halfway, and now you're way at the top. And then you keep secreting those for weeks. So now you're at week 5 and you're still secreting more than you ever secreted in the first instance. And that's going to continue to slowly level off until you get back down to normal levels. But for weeks and weeks, you'll be at high levels. Okay. And then again, the other part of why the secondary response is so cool, why vaccines are so great, is that you don't have to experience the actual illness. You have no symptoms, and you just get immediately so many antibodies that you're able to get rid of any and all of that pathogen and not get sick. Yay. Okay, so active immunity refers to you were actually exposed to the pathogen, like you inhaled somebody's respiratory illness, hopefully not COVID-19, but like a cold or a flu, right? And so now you have that pathogen in your body, and your body responds by making an army of B cells. They're also going to make an army of T cells, which we will talk about next. Okay. So that's a natural exposure to a cold, right? But you could also have an artificial exposure, which would be your vaccine, which is only antigen, no risk of getting sick, right? And it's active because it's going to stimulate your B cells and your T cells, but we really just care about the B cells when it comes to vaccines. So it's active because you're exposed to antigen, and it causes your adaptive immune system to respond, okay? Natural exposure will also cause your adaptive immunity to respond, but you also will probably be sick, right? Whereas with the vaccine, you won't be sick. Okay, so vaccines, it's very difficult. It takes a lot of time to make a vaccine, make sure it's effective and safe. So we choose diseases that have high mortality rate, or that cause so many severe illnesses that it is detrimental to the economy. So diseases with high mortality, or that have lifelong effects for the survivor. So like polio, crippled hundreds of thousands of children. They, of course, grow up to still be crippled. So that might be one or both legs, one or both arms, or any combination thereof, right? So polio didn't kill a huge amount of people. It did kill people, but the primary concern was the lifelong effect of not having use of one or more limbs, okay? So we, again, high mortality or complications, lifelong complications, or like the flu, where in addition to having a kind of high mortality rate, you also see just a lot of people get very sick and miss, a week or two of work. They could be hospitalized, etc., etc. Okay. Now, occasionally people experience a fever or tiredness from a vaccine, and that's due to activation of the immune system. Your body's working very hard to make that army of B cells, right? Very, very rarely a person is actually allergic to an ingredient, such as a preservative in the vaccine. Again, this is super rare. You would know from childhood, right, if this was the case. So adults do not need to be afraid of vaccines. And then parents, you don't need to be afraid of vaccines either. But, very, very rarely the child could have a reaction where you would be told that you need a special type of vaccine. You have to let them know ahead of time to order it so that it won't have that ingredient that the child's allergic to. Okay, vaccines cannot cause any of the crazy things people talk about, right? It cannot cause autism. That's a genetic disease. Vaccines cannot change your DNA. They are stimulating your immune system, and that is it. Okay. Passive immunity. This is when you're given antibodies. So your system, your adaptive immunity system was not stimulated, you did not produce these antibodies, and you will not have lifelong immunity. As soon as the antibodies wear out or used up, then they're gone. Okay. But this is quite normal, mother to child, right? And then sometimes in hospital settings for treatment. So antibodies given to a person, this could be from another person, or it could even be from an animal, like in a lab setting. Okay. So mother to fetus antibodies are small enough to cross the placenta. So babies are protected temporarily by their mother's own antibodies. Okay. Mother will keep passing some of these antibodies to the baby while she breastfeeds. So this is one reason that breastfeeding is very beneficial. Okay. And I think the largest amount of antibodies that are passed from mother to child are the first 1 to 2 breastfeeding events. So if mothers don't continue to breastfeed, as far as I understand, that's not really any harm to the child. Not breastfeeding at all does put the child at a little bit of a disadvantage, but we're really good at taking care of newborns, so it's not detrimental, but it's definitely preferred if you can breastfeed that first couple of days. Yeah. Okay. So this type of immunity, passing antibodies to from one individual to another individual, as I said, is temporary because the antibodies will be used up and then they're gone. You're not able to make any more because you didn't have the immune response to produce them in the first place. Okay, but this gives the baby time to get their immune system functioning so they can start making their own antibodies. Okay, so I mentioned T cells earlier, and now we're finally talking about them. So the other major player in adaptive immunity are T cells. We call this cell-mediated adaptive immunity simply because they're not producing antibodies. Okay, so why are they called T cells? Because they mature in the thymus. So they originate in the bone marrow, just like the B cells, but then they travel to the thymus. And again, this is stimulated with the hormone thymopoietin. You remember that? And this will provide specific immune protection, but without secreting antibodies. So T cells do not interact with free antigen. Okay, antigen has to be displayed to them. So sometimes I like to think of T cells as being kind of snooty. They want other cells to do some work for them first. And so this is where your antigen presenting cells come in handy, right? So B cells can present antigen, and other phagocytic cells such as monocytes mature into macrophages. So macrophages and dendritic cells. So what are your APCs, antigen presenting cells? Macrophages, dendritic cells, and B cells. Okay, so our T cells will read the MHC, which stands for major histone complex, or compatibility. And we have two types, 1 and 2. So MHC1 is on all of the cells of your body that have a nucleus. So your red blood cells don't have this, okay? But everything else in your body will be labeled with this MHC1, which we call MHC1 tag, okay? Or receptor, more appropriately, receptor. And then MHC2, only the cells of your immune system, so your white blood cells, they will have MHC2, okay? And so the T cell will know whether it's a MHC1 or MHC2. And depending on the type of T cell, this will matter. So let's look at that. So life of a T cell. Again, it begins as a stem cell in the bone marrow, and then it will travel to the thymus and mature. And this maturation process is to gain receptors, okay? And then it will travel to the lymph and the blood, mostly lymph. And if it ever comes in contact with an antigen, it will become activated. And this could be a helper T cell. This could be a killer or cytotoxic T cell. And it also could be regulatory or memory T cell. Okay, so those are the 4 types of T cells. Okay, so I'm not really going, I have 4 here, but any one of these, helper T cells, killer, cytotoxic T cells, and regulatory T cells, all of those can form memory cells. So I didn't count that as a separate. So regulatory T cells prevent autoimmunity. They're regulation, right? They're making sure that the T cells are following the rules. You're not going to attack yourself, and you're not going to overreact, okay? Helper T cells are your CD4 cells. Super important. Helper T cells are your CD4 cells. They make up 60 to 80% of all T cells, okay? So this shows you how important your helper T cells are. They become activated when antigens presented to them. And remember that our antigen presenting cells are your phagocytes, like your macrophages and your dendritic cells, and then also your B cells. When antigen is presented to the helper T cell, it will respond by proliferating into what we call effector cells and memory cells. And the effector cell, it performs an action, and for helper T cells, usually that action is to release these chemical messages to tell like a B cell to go ahead and proliferate and make its clonal expansion army, okay? But it can have different functions depending on what type of cell presented antigen to it. Okay, so activated helper T cells secrete cytokines, right, the chemical messengers, to help the cell that presented to it. Okay, so as I mentioned, if antigen was presented to it by a B cell, the cytokines are going to tell the B cell, yes, go ahead and make your army. If it was a cytotoxic T cell that is around, then these cytokines are going to tell the cytotoxic T cell to proliferate. And then you have another type of T cell are the CD8 cells, and that's the cytotoxic T cell. So cytotoxic T cells, aka killer T cells, they will detect bad cells. This is how we constantly kill cancerous cells or infected. Okay, so when a cell becomes infected and when it becomes cancerous, its self-tag gets funky. Okay, the MHC1 tag gets weird, and that's going to allow the cytotoxic T cell to detect it. So the cytotoxic T cells are constantly checking receptors on all of the cells that it comes in contact with, and it's making sure that the MHC1 tag is correct itself, and anything that has a weird tag or a foreign tag will be destroyed. So cytotoxic T cell will release granzymes to destroy the DNA of the bad cell, and it'll also release perforin to make a pore and cause the bad cell to lyse open. This should sound familiar. We talked about a different type of cell that has a very similar function when we talked about the innate immune system. Make sure you don't confuse the two, so go back and review that. Maybe make a compare and contrast chart. Okay, antigen processing. So the pathogen is engulfed by a phagocyte. That could have been a macrophage, a dendritic cell, or a B cell. And then that antigen will be displayed or put on the self tag. And since these are all white blood cells, the self tag will be type 2. And then that will be presented to a helper T cell. The helper T cell recognizes the MHC2 tag and that it's displaying antigen, and it responds. And if it was a phagocyte that presented, the helper T cell becomes activated. If it was a B cell, then you would need this T cell to have already been activated by that antigen on another cell. Okay, and then that's going to activate the B cell. And I know this is hard. This is tricky. Okay, so in order for a B cell to be activated by a helper T cell, the helper T cell would have already needed to be activated by a phagocyte. This is how we know that, yes, you actually have a pathogen in the body. You don't want to respond to something that's not actually a threat, right? So a check and balance is, this B cell that's become activated, did it correctly become activated? Is there actually a problem? Is this a correct antigen to be making antibodies against? So once a phagocyte has presented this antigen to a helper T cell, then that helper T cell is ready to activate a B cell that has also come in contact with that same antigen. This is a video to help review the process of antigen processing. Okay, and then here is a picture. So you have a white blood cell. It has the MHC class 2 molecule, and it is displaying a foreign antigen on it, and it's displaying that to the helper T cell. Previously, right, I'm sorry, back up. So I explained that, but I didn't explain what this cell is. So this is a macrophage. This is your phagocyte. It has come in contact with a foreign particle. It displays that foreign particle antigen on its MHC2 tag. A helper T cell comes around and reads that, oh, we have a phagocyte that has picked up a foreign antigen. Now he's on high alert or activated. Once that activated helper T cell comes in contact with a B cell who has also become activated to that same exact antigen, then the T cell will release cytokines to tell this B cell, yes, go ahead and proliferate, form your clonal army. Okay? So a phagocyte comes in contact with a pathogen, takes that pathogen up, processes it, displays it on the MHC2 tag. That gets displayed to a helper T cell. Now the helper T cell is activated, and if it comes in contact with a B cell that is displaying the exact antigen, then that B cell will be told by the T cell to go ahead and proliferate and form the clonal expansion, form the clonal army. Okay, so that is it. That's everything for the immune system. So a little review of the adaptive immunity, because I know it's a lot and it's difficult. And excuse my sloppy handwriting, I'm not used to writing on a computer. But you have your humoral. Okay, this is the formation of antibodies by B cells. B cells stay and mature in the bone marrow. You have trillions of B cells with unique receptors waiting in case they ever come in contact with their corresponding antigen. If they do, and they have the helper T cell, then they will form a clonal expansion army. Some of those become memory cells, some of them become plasma cells. The memory cells stick around to provide lifelong immunity, and then you would go back and look at that secondary response, right? Plasma cells are the effector cell. They immediately start to secrete antibodies. They'll do that for a few weeks, okay? And then and in low levels, they'll continue to secrete antibodies for a very long time. Okay, and again, in order for clonal expansion to occur, you have to have had activation by an activated helper T cell. Now, on cell mediated, you have T cells. T cells will move to the thymus to mature, and then they'll go back to the lymphatic system. You have helper T cells and you have cytotoxic T cells. The helper T cells are CD4. The cytotoxic T cells are CD8. CD8 requires a helper T cell as well to become activated. Then it will seek out and kill targets. Okay. They will also proliferate when they come in contact with the helper T cell. So it's not just one guy trying to go around and do all the work. Okay? Helper T cells will help your B cells, and they'll also help your cytotoxic T cells. And it helps them to proliferate. So without helper T cells, there's no proliferation and there's no big response, right? So helper T cells are like your check and balance that makes sure you don't get a huge immune response that's not actually necessary or correct. All right, I hope you've enjoyed our 2 lectures on the immune system. Email me if you have any questions.